Hello friends, myself Dr. Lily Dubey, and today I am going to teach this molecular basis of inheritance for the class 12 biology CBSE, and under which I am going to cover the topic transcription in eukaryotes. I have already covered various topics under it, and if you want to watch those topics, you have to subscribe my channel, and you will be able to watch it, and even you will be able to watch the forthcoming my videos on this molecular basis of inheritance. So let's start. Under this, I am going to cover the central dogma, types of RNA, transcription in eukaryotes, post-transcriptional modification, and few questions answered. So, what is central dogma? Central dogma was proposed by Francis Crick, and what does it mean? It means that the genetic information flows from the DNA to RNA and RNA to protein. So from replication, your DNA replicates, and from DNA through the process of transcription, mRNA is produced, and from mRNA on with the process of your translation, protein is produced. And what is then reverse transcription? Reverse transcription is genetic information flows from RNA to cDNA, and basis RT-PCRs are based on this cDNA only. So you need not to understand all this in details. Only for the class 12 level I am going to deal with. So what is transcription? It is a process of copying genetic information from one strand of DNA to into RNA is called transcription. That is DNA to mRNA. And in replication, total DNA is duplicated. But in transcription, only a segment of DNA or only one of the strands of DNA is copied into the RNA. That is the difference between your transcription and replication. So in transcription, why these both strands are not copied simultaneously? Means at the same time, why both copies are not copied. So there are two basic reasons for it. First is the genetic information transfer machinery would become very complicated. How? Suppose imagine the two strands are there, the strand 1 and strand 2 of the DNA, and both acts as template. Then what will happen? From both your messenger RNA will produce and different sequences of RNA, means one mRNA 1, your mRNA 1 will be produced, and mRNA 2 will be produced from the second strand. And finally, what will happen? Two types of protein will be produced, protein 1 and protein 2. Finally, it will be very complicated. So the whole mechanism will become very complicated. So naturally, the system tries to make the things easy going. So that's why only transcription, both strands are not copied simultaneously. And the second reason is that RNA would not translate into protein. Why? Because the two RNA molecules will be complementary to each other. Then what will happen? They will bind each other to form double stranded. And then finally, no translation of RNA into protein will take place. Now, what is transcription unit? Transcription unit is required in transcription. It is defined by three regions in DNA: promoter, the structure gene, and a terminator. Here is the promoter. Here is the transcription start site where transcription will start. This is a structure gene from which acts as a template strand or gene in the DNA. Here is a terminator, and here this is your coding strain where mRNA replaces T by U. Then what are different types of RNA and process of transcription? So different types of RNA are there: mRNA, tRNA, and rRNA. mRNA provides templates for transcription. tRNA brings amino acid and read the genetic code, whereas rRNA is structural and catalytic role during translation. But now we are focusing on mRNA because we need template for transcription. So how this transcription in prokaryotes and eukaryotes take place? There is basically some differences between both prokaryotes and eukaryotes. So what is the similarities? That in both the cases of transcription, your polymerase is required, DNTPs are required, and three steps of transcriptions are there. There is initiation, elongation, and termination. And difference is that in both the cases, in prokaryotes, polymerase 2 is only involved, whereas in eukaryotes, polymerase 1, 2, and 3 are involved. So what is the role of this tree? Polymerase 1, it transcribes your ribosomal RNAs. Polymerase 2, it transcribes precursor of mRNA, which is called heterogeneous RNA. And 3, transcribes tRNA and a small nucleate snRNA. So in brief, I will discuss about transcription in prokaryotes. So here in the figure you will see that I've already mentioned that there are 3 steps in transcription: initiation, elongation and termination. So here in the figure you can see that there is a sigma factor. Sigma factor is attached to this RNA polymerase. And when this sigma factor attached to the RNA polymerase, there is a site of initiation. Suppose this is your initiation site and this is your termination site. And somewhere here in between is your structural gene which you want, that part of the gene. So what happens, the sigma factor when it binds to the RNA polymerase, only then RNA polymerase starts transcription and then unwinding of the DNA starts. So as soon as the transcription starts, the sigma factor releases, come out, and then your transcription starts. It goes on. And at the last, at the termination point, your rho factor, it comes and attached to this your RNA polymerase. And then finally this rho factor, RNA polymerase and RNA, these three, all of them, they get separate. And finally we get in RNA transcript and this your DNA. So this is the transcription in prokaryotes take place in this way. So now in transcription in eukaryotes, we need only one strand of DNA duplicate its template strand is required. Its polarity is from 3' to 5', and second strand don't participate as I have already mentioned why, and it is known as coding strand, and its polarity is from 5' to 3'. In nutshell, what will happen, from DNA will get primary transcript as I have shown in case of prokaryotes, and then finally heterogeneous nuclear RNA, and finally this will go for processing. And after processing means this step splicing will be done there. After splicing, your matured RNA will be getting. So transcription in eukaryotes, let's discuss. In eukaryotes, structural genes are monocistronic in nature. Only one cistron is there, and splicing is there due to presence of your introns and exons. So in as I have discussed in prokaryotes, we get here your transcripts of RNA, which is known as heterogeneous RNA. Then it is having your exosplit gene, which consists of your exon and intron. Now these exons are coding sequence of DNA, which will give you protein, whereas introns are non-coding, which are not coding for anything. So what we have to do, we have to remove this intron from this RNA transcript, only then we'll get the final protein. So as I have told, the exon, they are express sequence and they have genetic information protein synthesis. But in this intron, there is no information protein synthesis is there. So how this processing take place? Now you can see here that when we get this RNA transcript, you will see here this is your blue is your mRNA, and also introns are there. This is your introns and this is your exon. So it consists of your introns and exons. Then there will be your capping will take place. Here it is your capping, that is your capping, and then polyadenylation will take place. Now how this happens, how this capping and polyadenylation take place. So as our target is to remove this introns and exons are joined in a definite way. So how this will happen? So tailing means it is 200-300 residues of your adenylated acids which are attached to 3' end of the this heterogeneous RNA. That's why the process is called polyadenylation, and it means many adenylic acid is attached to this 3' end. And in capping, and your unusual nucleotide known as methyl guanosine triphosphate, which is read as MGPP, is attached at the 5' end. So what happens now? Very important process take place, that is RNA splicing. Now in RNA splicing, these introns will be removed, and then all the exons will come together, and finally your matured mRNA will be produced. That is you can see here in the figure, and it will be having also a tail of A, poly A tail will be there at the 3' end. So in this way your splicing take place and matured RNA is produced. Means